What I wanted to do this evening, rather than talk and give you a potted history of the various things that I've done, as Neil indicated, it might not be a very linear or easy story to follow. I'm going to focus rather on one particular question, which I think is, or one particular topic, which I think is really quite fascinating and one which we still don't have the answer to. So I'm not going to give you answers, but hopefully, or the ultimate answer, I'll give you some answers, but also hope to give you some questions that maybe you'll want to go away and think about and we can talk about in the future. And the topic I want to talk about is oral vaccines and how we can make oral vaccines work better in the children who need them. And I'm going to particularly draw on experience from working on poliovirus for the last 7 or 8 years, although I will mention a couple of other oral vaccines. Okay, so the enormous attractiveness of oral vaccination is that it's very easy to administer an oral vaccine. You just need to give a couple of drops of the vaccine in the mouth of a child, and that child is protected. And in the case of polio, that child is protected for life from paralysis by a very nasty virus. So this is a picture of a child receiving polio vaccine in northern India during an immunization campaign. And I've asked my kids to give out a few vials of vaccine to you all. Don't worry, it's actually Ribena, not vaccine, because we're not allowed to use this vaccine anymore in the UK. But if you want to, you can put a dropper on the top and have a play around with how that works. It's very simple. My kids can immunize you. It's a very simple way of giving a vaccine. So as mentioned, this vaccine the polio vaccine has had a real impact on cases of paralysis due to polio. So in 1988, the world was a place where many children became paralyzed by polio, around 350,000 children each year. And many countries, shown here in red, had never managed to get on top of their polio problems. So all these countries were reporting cases of polio. And at that time, the governments of the world committed at the World Health Assembly to try to eradicate polio and to try to do that by the year 2000. And the rationale behind that was that this is a virus that only infects humans. So if you can eradicate the virus from humans, there are no reservoirs for infection. And in theory, we can go to a world where there are actually no more polioviruses in the world. The program made enormous progress. In 2002, there were just 7 countries which had not stopped poliovirus transmission using that vaccine. And there are a number of countries that became polio-free but were continually reinfected by movement of virus out of these endemic countries. So and as you see, in 2002, there were approximately 2,000 polio cases in that year. So a huge reduction, but of course the goal of eradicating poliovirus and paralytic polio by the year 2000 was missed. 10 years later, in 2012, we're almost there. So there are just 3 countries which have not eradicated polio. Those are Nigeria, Afghanistan, and Pakistan. And this year, the lowest ever reported number of cases globally, just 60 cases reported to date. So enormous progress, although there is some way still to go, and I'll say a little bit more about that. So these are some children with paralysis due to polio. And you can see this characteristic flaccid paralysis, which typically affects one limb And it means that these children will always need to walk with an aid for the rest of their lives. The virus does not paralyze every child it infects, so it only paralyzes around 1 in 200 of those children who are infected. Now, when we first started to look at this polio in my research group 7 or 8 years ago, as I say, there was a curious problem, and this was this. When we looked at the number of doses of vaccine that these children who were paralyzed in India had received, we saw that many of them had received multiple doses of that oral poliovirus vaccine. In fact, the majority had received more than 10 doses of vaccine. So something clearly was not right, something was not working. So in this talk, I'd like to cover these 3 areas. First of all, I'd just like to say something about the promise of oral vaccines, the promise they hold for the future for the prevention of disease. Secondly, I wanted to talk about why oral vaccines fail, and particularly focus on experience with polio. And thirdly, talk a little bit about how we might think about making these vaccines work better in children who need them. Okay. So just to take a broader context, the importance of infectious diseases. Infectious diseases are the leading cause of death in children under 5 years of age. Approximately 6 million children die each year as a result of infectious diseases, the majority associated with either pneumonia or diarrhea, but also other infections of importance here. I should say, though, that there is progress. So this is the death rate, the mortality rate in 1990, shown in these pale blue bars here. And with the dark blue bars, you can see the death rates in 2010. So you can see that they've really come down. And in some areas, they've come down really quite remarkably. The Millennium Development Goals, agreed at the UN, aim for a reduction by 2/3 of this mortality rate. And that's shown by these orange bars here. And you can see that whilst progress is being made, those goals, which we aim to reach in 2015, are still a little way off, particularly in sub-Saharan Africa. Now, obviously vaccines are not the whole story by a long way, but they do play a role. So this just shows the impact of the measles vaccine over the last 10 years. This blue line here shows the number of deaths each year due to measles, and you can see it's come down as vaccination coverage has improved. This dashed red line here is the number of children we would expect to die as a result of measles infection in the absence of vaccination. So these pink bars show the numbers of deaths averted through measles vaccination. So you can see that in 2010, around 1 million deaths due to measles were averted through the use of vaccination. So oral vaccines are part of that story, and you saw the progress for polio. There are also vaccines licensed for rotavirus, which is a major cause of diarrhea and associated deaths in children in lower income settings, cholera, and typhoid. But you might say, well, that's not really very many, and that's true, and I'll come on to why there perhaps are not as many oral vaccines as we would like to have. However, there are some more in the pipeline, mostly against intestinal infections, again, important causes of mortality and morbidity associated with diarrhea, but also that there is work towards looking at ways of giving vaccines for non-intestinal infections or non-mucosal infections by the oral route. So there is work on things, for example, malaria, oral vaccines for malaria. Okay, so the advantages of oral vaccine, I think, well, the first advantage is summed up by this picture, really. Here's a child receiving an injected polio vaccine, and here's the nice drops of oral vaccine being put on a sugar cube, which is all very pleasant. So they're, of course, well tolerated by children and accepted by children. They're easy to give. There's no need for medical staff, so they can be given by untrained volunteers. And, of course, there's no needles, so there's no problem with disposing of sharps. So I just want to talk about some of our experience working on poliovirus. And to do that, I want to just give a little bit of background for those of you who are not familiar with this virus. So this is a representation of the virus particle constructed on a computer. And obviously it looks big here, but it's only 3 millionths of a centimeter across. So it's a very tiny little virus, a icosahedral structure. When you're infected with this virus, at your peak infectiousness, you shed about 1 million of these per gram of your poo. So you're really highly infectious. And so this virus is spread typically through what's called the fecal-oral route, so through fecal contamination of food or ingestion of virus through contact with fecally contaminated objects. It may also be transmitted from mouth, directly from shedding in the mouth onto hands and into mouths. So that's the oral route of transmission. Shown here is poliovirus serotype 1. So there are 3 serotypes of poliovirus, and they all look slightly different in their structure. And so therefore they're seen differently by the human immune system. And so to get immunity against polio, you need to induce immunity against all 3 of those serotypes. So the body sees those as different viruses. So the oral vaccine, attenuated vaccine, was developed by Albert Sabin in the 50s and licensed in the early 60s. And the way that was done was by taking this virus found in the wild, and which causes paralysis, as I said, in about 1 in 200 infected individuals, and growing it, first of all infecting monkeys, collecting the virus and then infecting monkeys again, and then doing the same thing in cells isolated in culture from monkeys, and repeatedly doing this until the virus was adapted to grow in monkey cells and not in human cells. And at the end of this process, which is a very sort of empirical process for each of the 3 serotypes, Sabin generated what are now called Sabin polioviruses, type 1, 2, and 3. And these viruses are much less neurovirulent, so they're much less nasty viruses. So these viruses will replicate in your gut, they will induce immunity, but they're very unlikely to induce paralysis. So these viruses only cause paralysis in around 1 in a million infected individuals. So here's a vial of trivalent vaccine, which is what you've all been, or some of you will have been given. Typically this trivalent vaccine, the trivalent vaccine contains all 3 serotypes of poliovirus. So all the 3 serotypes are combined, and then that vaccine is administered to induce immunity against those 3 types. Now, when we look at how immunogenic that vaccine is, in other words, whether that vaccine induces protective antibodies in individuals, when that vaccine is given to children in lower income settings, we tend to get there tends to be not perfect immunity. So this is the degree of seroconversion, or the proportion of children seroconverting after 3 doses of this trivalent oral polio vaccine. And this is an average taken across 32 studies in lower income settings. And so what you'll see is that just over 20% of children, even after 3 doses of this vaccine, which is the standard course in the West, still do not have antibodies and so are not protected against polio. And what you'll also notice is that serotype 2 vaccine poliovirus seems to induce a better response than types 1 and 3. In fact, what we know now is that serotype 2 Sabin serotype 2 virus is a fitter virus. It will grow better in the gut, and it will outcompete the other two serotypes. And so the trivalent vaccine actually works reasonably well against serotype 2, but less well against serotypes 1 and 3. So if we look at the current picture, this dot map shows where all the polio cases have been reported globally in the last 6 months. And these red dots show serotype 1. So this is cases of paralysis associated with polio, wild poliovirus serotype 1. And these blue dots show cases of polio associated with serotype 3 virus. And you'll see that there's no serotype 2 virus. And that's because with the use of this trivalent vaccine, the Global Polio Eradication Initiative was able to eradicate serotype 2 in 1999. The last case was reported in northern India at that time. Okay, so when we first got involved in looking at polio, this was Christoph and myself. We wrote to David and to Bruce Aylward at the WHO and said we thought that there were some interesting questions that maybe we could look at the polio surveillance data and use some of our maths and stats skills to try and answer those questions. And the question that David and Bruce raised at the time was why was polio persisting in India? So at that time, you can see there was a lot of poliovirus serotype 1 and serotype 3, and in particular in these northern states of Uttar Pradesh and Bihar. And what was curious was that this was happening despite efforts of the program to focus their immunization, so vaccination campaigns, on children living in those northern states. And this just shows the numbers of doses, the average number of doses received by children under 5 years old over time between 1997 and 2000, and the end of 2005, beginning of 2006. And you can see, and these black lines here show that national immunization days, so these are campaigns where the whole country aims to immunize, all children under 5 years of age are immunized nationwide. And these green lines, which perhaps are a bit less clear, are sub-national immunization days targeted at particular parts of the country, and in this case very much focused on these northern states. And what you can see is that with increasing frequency of these campaigns, the numbers of doses of vaccine received by children in these, particularly in these northern states shown by the red line here, really reached quite high levels. So by the end of 2005, around 15, an average child under 5 years of age had received 15 doses of vaccine. So what we were able to show was that using a case control approach, so what we did here is we looked at the numbers of doses of vaccine received by polio cases and compared them to the numbers of doses of vaccine received by children who did not develop polio. Now, if polio vaccine is working well, then we'd expect the children who are cases not to have received very many doses of vaccine as compared to the controls, the children who were protected. And using that kind of approach, what we found was quite surprising. So we found that the efficacy of the trivalent vaccine, here shown against paralysis due to serotype 1, was really very low. It was only about 10% per dose of vaccine. So each time you gave a dose of vaccine, there was only around a 1 in 10 chance of the child developing protective immunity. And importantly, what we found was that the protective efficacy was lower in those northern states compared to other parts of India. So it appeared that failure of the oral vaccine, rather than the failure to immunize children, was the cause of polio persistence in India at that time. And so we were able to say to WHO that any efforts to improve vaccine efficacy would outweigh the benefits of further increasing the frequency of vaccination campaigns. And that, of course, WHO knew something was going on, and they were already developing a monovalent oral poliovirus vaccine. So that's a vaccine where you only include one of the serotypes in the vaccine vial. So using a monovalent vaccine, a monovalent vaccine was introduced in 2005, around the same time we were doing this work. And what we were able to show then was that soon after that vaccine was introduced, that it was almost 3 times more effective compared to the trivalent vaccine. And that's essentially a result of removing the serotype 2 and serotype 3 vaccines. So there's no longer competition between these strains, and immunity is, and the serotype 1 vaccine virus is much more likely to replicate and induce a vaccine, and induce immunity and protection against paralytic disease. So in other words, what was now possible? So this just shows the number of doses of vaccine, and this shows the proportion of kids who are protected against polio. And that you can see for the trivalent vaccine, to get just over 80% of children protected was taking 15 doses of vaccine. With the monovalent vaccine, the same could be achieved with just 5 doses. In 2005, a bivalent oral vaccine was licensed. In 2009, a bivalent oral vaccine was licensed. And this shows the immunogenicity of that vaccine based on a trial conducted by Roland Sutter and colleagues in India, in South India. And this shows the proportion of children after 2 doses of vaccine who developed protective antibodies for the standard trivalent vaccine, for the bivalent vaccine, and for the monovalent vaccine. And as you can see, this bivalent vaccine, which contains both serotypes 1 and 3, but not serotype 2, is almost as good against serotype 1 as the monovalent vaccine, and similar results for serotype 3. So this bivalent vaccine will protect against both serotypes 1 and 3, and it's through the use of these vaccines and in addition, identification of migrant kids who were unimmunized that polio was eradicated in India. And this is the last case of polio was reported in the beginning of 2011. And this is Rukhsar Khatun. She's the child who was last paralyzed by polio in West Bengal. And hopefully the last child paralyzed by polio in India. There's always a risk of reintroduction. So I'm going to take my jacket off because I'm roasting. Okay. Okay, so this is, oops, I'm still connected to my jacket. Sorry. Okay. So this is obviously a great success story for the Global Polio Eradication Initiative. But it was an effort. The Indian government was spending close to $200 million a year. And the average number of doses received by children under 5 years of age in northern India was around 19 at the time that polio was eradicated. And indeed, by the age of 4, children had received on average 29 doses of oral vaccine. And this problem with the oral vaccines not being as immunogenic as we would like is not restricted to the polio vaccine. So this shows antibody responses, protective antibody responses to rotavirus vaccine, which protects against rotavirus diarrhea. And here you can see the average immunogenicity of this vaccine recorded by its manufacturer, GlaxoSmithKline, according to the World Bank Country Income Group. And you can see a decline in the geometric mean concentration of antibodies following 2 doses, which is the standard course for this vaccine, as you go from high to upper middle to low income settings. So the same problem has been observed with rotavirus vaccine, and that translates, of course, to reduced efficacy of that vaccine in these in lower income settings. So this just shows vaccine efficacy as a function of the gross domestic product of those countries where the vaccine has been, where vaccine trials have taken place. And what you can see is, as you go down to countries with a low GDP, then you can see a low vaccine efficacy. Now, in the case of rotavirus, it's a little bit more complicated story because, of course, there are different genotypes and because it's a disease of the intestine, so there's some quite complex immunology there. So it's a little bit more than just the immunogenicity of the vaccine. But for polio, it's all about the immunogenicity. If you can induce immunity, any level of antibodies in the blood, then a child is protected against paralytic disease. Okay, so why might these vaccines fail? Well, there have been a number of ideas around for a while. We know that maternal antibodies in newborns and antibodies, IgA in particular, immunoglobulin A, received through breastfeeding, can inhibit vaccine virus replication. But the practical implications don't seem to be there. So if you give a vaccine to a neonate, an oral polio vaccine to a neonate, it seems to be, in general, quite immunogenic and quite effective. And indeed, many countries include a birth dose of oral poliovirus vaccine, in their routine vaccination schedules. It has been hypothesized that malnutrition or deficiencies in micronutrients, and particularly vitamin A or zinc, might contribute to the failure of these vaccines. There have been a number of trials that have looked at giving vitamin supplements, vitamin A and also zinc supplements, to try and improve the immunogenicity of polio vaccine and also other vaccines. Sorry, I should have mentioned that cholera and typhoid vaccines also show the same problem. And those trials have had mixed results. Some have found no effect. There's been a small number of trials which suggest a small effect, but not really that significant in terms of having a real-world impact. There may be a role of human genetics, so people differ in their immune systems as a result of the underlying differences in their genetics and HLA types and so forth. That doesn't seem to be important because it doesn't correlate with where we see these vaccines fail. And then finally, a high prevalence of other intestinal pathogens may interfere with the response to these to oral vaccines. And there have been mixed findings, but for a number of studies have found that in children who have got diarrhea at the time of vaccination, they're less likely to respond to the vaccine. So one way of looking at vaccine response is by measuring virus in stool. If you give a child vaccine poliovirus, it will replicate in their intestinal mucosa, and virus replication is what's needed to trigger the immune response. So you give this attenuated virus, it replicates, it doesn't cause disease, but it's enough for your body to mount an immune response. So if the virus induces a response, in other words, if there's vaccine, what's called take, then we would expect to see vaccine virus shed in stool. We did a study with the Enterovirus Research Center in Mumbai. This was with Dr. Jagadish Deshpande, and they routinely test for vaccine poliovirus in stool samples collected from hundreds of thousands of children with acute flaccid paralysis in India. Most of those children have paralysis for causes unrelated to polio. And so what we were able to do was to look at the detection of vaccine poliovirus in the stool of children as a function of when they were immunized. And what we found was is shown here. So what this graph, this is a slightly confusing graph, but what we have here is the months of the year going round from January through to December. And then this black line here indicates the relative odds of detecting vaccine virus in the stool compared to April. So this is set at 1 for April. And what you can see is that this really shows very strong seasonal variation. So in the winter months, you have a good chance of getting poliovirus shedding after administration of the vaccine. But in these summer months, during the monsoon, you see really very low levels of shedding after administration of the vaccine. And interestingly, that's the opposite to what we see when we look at prevalence of enteric pathogens in the stool. So this shows the relative prevalence of non-polio enteroviruses, in this case, in stool samples collected from children in India. And so you can see that the isolation of these enteric pathogens and isolation of vaccine virus occur at different times of the year. It suggests that enteric viruses and other enteric pathogens may be important for the reduced response to the vaccine during the summer. That's obviously only suggestive. We don't really have an answer directly from that, but it's very suggestive that there's something seasonal which affects the response to the vaccine, and it's quite likely to relate to the prevalence of enteric pathogens, which are themselves strongly seasonal. So I don't really want you to take this slide in, but this is our sort of working hypothesis for what's going on. Bacterial and viral pathogens and parasites are stimulating an innate immune response. So that's the first line of defense in the intestine, which can be cross-protective. So it's not a specific adaptive response, but it's cross-protective against other pathogens in the gut. And in particular, we think that innate immune effectors such as type 1 interferons, natural killer cells, induce a resistance of the intestine to replication of your vaccine poliovirus. So if you've been exposed to a lot of pathogens, your gut can become inflamed. You can have activation of all this innate immunity, which is effective at protecting you against other pathogens, but it's also effective at protecting you against replication of the vaccine virus. And therefore, this vaccine doesn't induce an immune response. It doesn't replicate sufficiently to induce an immune response. So that's our hypothesis, and now we're testing it in a clinical trial. So what we're doing in that study, which is joint with Cherry Kang, Jacob John, and others at the Christian Medical College in Vellore, and then other collaborators both at the University of Oxford, at Liverpool, and of course at Imperial, is to treat. I'll describe the study. What we're doing is we're taking children, infants 6 to 11 months old who lack antibodies to polio, and we're taking half of them randomly and treating them for their enteric infections using antibiotics. We're using a 3-day course of azithromycin, and we're also using an antiprotozoal, so that's a 3-day course of nitazoxanide. And in the other arm, we're not providing any kind of treatment for enteric pathogens. And 2 weeks later, we're immunizing them with monovalent poliovirus vaccine. And we're going to compare the vaccine response in each arm to see whether treating enteric pathogens improves the response to the vaccine. And in doing this, we will also be measuring the prevalence of these enteric pathogens and markers of activation of the innate immune response and intestinal inflammation. So we will get some interesting information, not just about the prime outcome, but also about the association of these of specific enteric pathogens or markers of innate immune activation with vaccine failure. Okay, so that's a study that's just beginning, and we're hoping to start recruiting in October. We have ethics approval, so if you come back in 3 years, hopefully I'll be giving you a lecture with some of the results from that study. So I wanted to move on to another aspect of giving vaccines by mouth. And for that, I need to talk a little bit about the immunology, but really very little, because I'm not an immunologist, so please don't ask questions about this part. When vaccine is taken up, or antigen is taken up, it's processed by antigen-presenting cells. So these are dendritic cells or macrophages, and it's presented by these cells in the body in lymphoid tissue to the immune cells, the T helper cells, the T cells and the B cells. The virus is presented to T helper cells, which then process that vaccine antigen and will activate B cells which recognize the same antigen, so they're cognate. It's a cognate system, and these B cells develop into something called plasma B cells, and they produce antibodies which will neutralize the virus, and they're what will protect you against disease and clear infection. Now, the fact that you give a vaccine by mouth means that it gets processed by these antigen-presenting cells in the intestine, in fact, in the gut-associated lymphoid tissue. And there's something special happens there, and that is that these antigen-presenting cells release something called, or release an enzyme that produces retinoic acid. And retinoic acid acts on the T cells and the B cells to induce receptor molecules on their surface and integrin molecules. So these are protein molecules on the surface, CCR9 and alpha-4 beta-7, which guide these cells when they circulate around your body to home to the small intestine. So the fact that the vaccine was given by mouth and was processed by these antigen-presenting cells in the intestine means that the immune response happens in the intestine. And then, of course, memory cells are produced, and these were what protect you against disease and infection in the future when you next come across that virus. Retinoic acid also is important for something known as class switching, so it's important for causing these the type of antibody to be produced to be this particular type IgA, which is secreted into the intestine and neutralizes the virus in the intestine, and also can play a role in extruding the virus across the endothelium as well. So the fact that antigen or vaccine is first encountered in the intestine is very important for the localization of the vaccine response in the intestine. So what that means is when we compare the oral vaccine with an injected killed vaccine, so this shows this is antibody titers with days after vaccination, and you look and see the killed injected vaccine are shown by these dashed lines here, and the live oral vaccine is shown by the solid lines here. And you can see that both vaccines induce neutralizing antibodies in the blood, but only the oral vaccine induces antibody in the small intestine. The killed vaccine or the injected vaccine doesn't induce any kind of antibody in the small intestine, any kind of IgA in the small intestine. Why is that important? Well, for intestinal pathogens that cause diarrhea, it protects against diarrhea. But for poliovirus, it's important because in an eradication program, we want to reduce transmission. And so we rely on something called herd immunity. So I apologize for this picture. Here we have a child producing some feces into the environment. And if you have a vaccine that induces antibodies in the intestine and reduces poliovirus replication and shedding, then that means less poliovirus in the environment. And that means even if you're not immunized, you're less likely to encounter the virus. And so that's what epidemiologists like to call herd immunity. So you don't need to vaccinate everyone to eradicate an infectious disease from the population if you can reduce the amount of virus shed into the environment. Okay, so one way of measuring this kind of intestinal immunity is, after you've given a vaccine, then to give another dose of the vaccine poliovirus. And this is kind of like a challenge. So if this individual has, after vaccination, has developed an immune response in their intestine, then the next time you give them vaccine poliovirus, they should be protected, and therefore they shouldn't shed the virus. So the next time you give the virus, rather than wanting to see shedding, we'd expect not to see shedding because they've got mucosal immunity in the intestine. So when we looked at this, we did indeed find that as you increase the number of doses of vaccine received prior to that challenge, then you see a reduction in the odds of shedding that challenge poliovirus. But what you can also see here is that even after a lot of doses of vaccine, children still have a reasonable odds of shedding virus. So the protection is not complete. So it's not sterilizing immunity. And what we recently found also was that this immunity wanes over time. So this shows the relative odds of shedding challenge virus for each of the 3 poliovirus serotypes as a function of the time since they were last vaccinated. And so you can see the odds of shedding virus increases as you increase time since the child was vaccinated. And so that suggests quite rapid waning of intestinal mucosal immunity to poliovirus. Now that might be important because what it means is that kids who have been vaccinated could still become infected with the wild virus, and so they could still take part in transmission of polio, and that makes eradication that much harder. And indeed, when we looked for wild virus in stool samples collected from healthy children who had been immunized, but who were, and in this case, children who received more than 9 doses of vaccine, but who were in contact with children with poliomyelitis, then what we found is quite a few of those children, despite being immune, were shedding wild poliovirus. This is serotype 1 and serotype 3 here. So these children, even if they've been immunized with OPV, can participate in transmission as a result of waning mucosal immunity. Okay, so the other study, again, which is, and this is, well, there's the 2 studies, one of which has been completed and one of which is about to begin, which we are involved in, aims to try and boost mucosal immunity. And it aims to do that not by giving additional doses of the oral vaccine, but by giving a dose of the injected killed vaccine. So whilst, as I showed you, the killed vaccine does not induce any kind of intestinal immunity in children only vaccinated with that vaccine, in children who have previously received the oral vaccine, these children appear to be what's called mucosally primed. So they've already got those homing receptors on their T cells and their B cells. And so the next time that you give them an injected vaccine, you can boost their immune response, and you get a response not just systemically in the blood, but you also get a response in the intestine. Now, the advantage of using the injected vaccine is that, unlike the oral vaccine, it doesn't have this problem with reduced efficacy and immunogenicity in lower income settings. So in these 2 studies, what we're trying to assess is whether an additional dose of IPV, the injected vaccine, or OPV, compared with doing nothing, reduces shedding of challenge poliovirus given a month later. And as I say, there are 2 studies that we're involved in. One has taken place in Moradabad, and that was very much led by WHO and by Dr. Jagadish Deshpande at the Enterovirus Research Center in Mumbai. And there's another study which we're also closely involved in South India, which is led by Dr. Jacob John at the Christian Medical College in Vellore. The results are just coming in from this first study, and they seem to be quite encouraging. So what they suggest is that an additional dose of IPV may be an effective boost for intestinal immunity, and therefore this vaccine might have a role in the final stages of global eradication, given perhaps during immunization campaigns to children who were last immunized with oral vaccine sometime in the past. So perhaps the kind of settings where we might think about using this vaccine would be places like, for example, Pakistan, where access to children can be intermittent because of problems with conflict and security. Okay, so this is my last or penultimate slide. What's the future of oral vaccines? And so what this picture was taken from this month's copy of Scientific American, and it's a little bit far for you to see, but what it tries to illustrate is all the microbes, all the bacteria and viruses which are associated with our body. And of course, the intestine harbors many millions, many billions of bacteria and viruses. And I think, and as we've seen, some of these can interfere with the response to oral vaccines. So a lot of future progress in oral vaccination will come from a better understanding of the interactions of this, what's been called the gut microbiome, with the vaccine, and of the vaccine and microbiome with the innate immune response, and how that then ties into activation of the adaptive immune response. So I think progress will come there. Progress, I think, will also come from better understanding how our immune cells are targeted to specific organs in our body. So you saw that T and B cells stimulated in the gut develop this gut homing phenotype. And there's been some very exciting recent work that looks at how you could give an injected vaccine with retinoic acid in this example, or with other so-called adjuvants that result in a response that, whilst you're giving the vaccine in the arm, induces immunity in the intestine. Okay, so thank you very much for listening. There's a few acknowledgements just in particular for this work that I've talked about today. For the trial in India, the trials in India, I'd particularly like to thank Cherry Kang, Gagandeep Kang, and Jacob John at the Christian Medical College in Vellore. I'd especially like to thank colleagues at WHO who I've worked with closely now over the last 7 or 8 years, especially Bruce Aylward and Roland Sutter. I would like to thank my research group, who obviously are critical to a lot of this work and to a lot of work which I haven't described. Kath O'Reilly, Isabel Blake, and Tara Mangle. And I'd like to thank other colleagues at Imperial who have been involved in the polio work, Crystal and Christoph. Now, of course, many other people I would like to thank, and I haven't put on this slide. And I can see that my son is yawning now, so that's a good sign. So I'd really like to thank all of you that I have had discussions about this work with, and I have worked with in the past. And also, of course, I need to acknowledge people who have funded this work.